Hi everyone, I'm Theo. I'm currently a PhD student at Imperial College London and Kew Gardens, but today I'm going to talk to you about my master's project that I did at the NHM in London. And it was on molecular phylogenetics and biogeography of caecilian amphibians. So first of all, what is a caecilian? So they're an order of amphibians that forms the sister clade to the frogs and newts. They're relatively diverse, comprising of 10 families and around 200 species. And they're characterized by the independent evolution of limblessness. So no, they're not snakes. They have a cryptic lifestyle and a tropical distribution, meaning they're the most poorly understood amphibian order. So African caecilians comprise a substantial portion of the group's diversity, hosting 4 of the families. And the species limits in these groups are relatively poorly understood due to the similar external morphology between species. So it's therefore important to investigate the molecular diversity within these groups. And here we investigate the genus Scolecomorphus within the family Scolecomorphidae. This genus is endemic to the Eastern Arc Mountains, which is a biodiversity hotspot and a center of endemism. So Scolecomorphus currently contains 3 species, but no molecular analysis has been done on this group, and Scolecomorphus kirkii is believed to comprise multiple species. So as we know, many molecular phylogenies rely on mitochondrial data, but we're aware of the issues surrounding this, such as incomplete lineage sorting and introgression, which can lead to gene tree, species tree discordance. So here we're using multiple unlinked loci from nuclear and mitochondrial markers to generate the phylogeny. So using museum specimens, we're aiming to characterize the molecular diversity of the genus, determine the distribution of genetic variation among the species and the Eastern Arc Mountains, and test whether the current taxonomy holds up. So this is the first tree. So although we said about combining nuclear and mitochondrial data, we weren't successfully able to amplify any of the nuclear markers during my master's, so we're currently working on that now. So I'm presenting only the mitochondrial phylogeny. Here's the Bayesian phylogeny, but the maximum likelihood in Bayesian phylogeny is broadly agreed. And the key takeaway here is that there's a lot of diversity within Scolecomorphus kirkii. So although Vittatus and Uruguayensis are monophyletic, kirkii clearly isn't. And so this is supported by our species delimitation analyses, which suggests there are at least 10 species level lineages within Scolecomorphus. So looking at the biogeography, the broad scale biogeographic patterns show an isolation by distance effect, with many of the clades also being restricted to specific mountain blocks. But we have to take some care here because we can see that the relatively low correlation coefficient of the distance correlation suggests that there's a more complex biogeographic history. And this is shown here. So we can see that many samples collected from the same mountain block. So for example, the Udzungwa samples, although they're collected from the same mountain block, they're polyphyletic. So this suggests a complex biogeographic history and potentially several unique species occupying the same mountain block. So finally, what are the next steps? So we're going to incorporate the nuclear DNA sequences from all the samples, and then hopefully use this to describe additional species through an integrated taxonomic approach. And then finally, and importantly, assess the conservation status of these newly described species. So yeah, I just want to say thank you to the whole team, and please connect with me on Twitter or email, and also the students committee. So yeah, if anyone has any questions.